To all the NEET aspirants, this is your Prasanna Ma'am from NEET World, and yes, we are going to start our major chapter, that is molecular basis of inheritance, all the NEET previous year questions. Along with the concepts, we are going to exercise beta. So without much delay, let's get started with the first question. So here if you see, first question which is asked in the year NEET 2025. In the year NEET 2025, only from the chapter molecular basis of inheritance, more than 5 questions have raised out. So that much important chapter it is. So listen and focus on each and every question with maximum interest. Okay. So first question, if you have to look into beta, which is match the following type of question, wherein on the left hand side we have the scientists and the chromatins, and on the right hand side what type of chromatins and what experiment they have done is given. We have to match it. First is Alfred Hershey and Martha Chase. They have given an unequivocal proof that DNA is the genetic material. So A is option 4. Euchromatin and heterochromatin. What is euchromatin, which is active and loosely packed chromatin, is called as euchromatin. So euchromatin is loosely packed and lightly stained. This is active chromatin. Heterochromatin is inactive. It is densely stained or dark stained, which is option 2. Next, Frederick Griffith. Frederick Griffith has worked out on the experiments, has worked out on transformation principle. He has conducted experiments on the bacteria Streptococcus pneumoniae. Okay. So here if you have to look into the right option, A is 4, B is 3, C is 1 and D is 2. Option 1 will be your right answer. Next question you look into match list 1 and list 2. Adenosine. Adenosine is what? Nucleic acid. Adenosine is what? Nucleoside. That is nitrogen base plus sugar. Okay, nitrogen base plus sugar. That is adenine plus ribose sugar. Adenine plus ribose is adenosine. It is a nucleoside. So A is option 3. Next, adenylic acid. Adenylic acid is what beta? Nucleotide, which is nitrogen base plus sugar plus phosphate group. Nitrogen base plus sugar plus phosphate group, which is a nucleotide, which is option 2. Adenine is a nitrogen base, which is option 1. Alanine is an amino acid, which is 4. So here if you have to see the right options, A is 3, B is 2, C is 1 and D is 4. Option A will be your right answer. Next question you look into again NEET 2025, statement based questions, multi-statement based questions. Which of the following are the post-transcriptional events in a eukaryotic cell? First you recall what are the post-transcriptional events that you come across in a eukaryotic cell. 3 post-transcriptional events. One is 5' capping, 3' polyadenylation, 3' polyadenylation, and third is RNA splicing. These are the 3 post-transcriptional events that happen in a eukaryotic cell. These post-transcriptional events occur inside the nucleus itself, nucleus of eukaryotic cell. Now you look into all the statements Transport of pre-mRNA to cytoplasm, prior splicing, no, this is not a post-transcriptional event and this will not happen at all. B is what? Removal of introns and joining of exons. Yes, this is RNA splicing, this happens during this is a post-transcriptional event. C is addition of methyl group at 5' end of HN RNA, heteronuclear RNA. Yes, that is 5' capping. So option C is right. D, if you see, addition of adenine residues at 3' end of heteronuclear RNA, D is right, because many adenine groups will be added at 3' end, which is polyadenylation. Base pairing of 2 complementary RNAs, this will not happen. This is a not a post-transcriptional event. So here if you see, what are the right options beta? B, C and D. Option 4 will be your right answer. Look into it again, statement based question in the same year NEET 2025. Given below are two statements. In RNA world, RNA is considered as the first genetic material evolved to carry out the essential life processes. RNA acts as genetic material also as a catalyst for some important biochemical reactions in living systems. Being reactive, RNA is what? Unstable. RNA is unstable as it is reactive. True? Yes, in RNA world, RNA is considered as first genetic material, true, and RNA acts as genetic material, it acts as a catalyst, it also acts as a structural component in ribosome, and it helps in biochemical reactions in living. It is important for biochemical reactions in living systems. It is reactive and unstable because it has OH group, free oxygen group at 2' end in the ribose sugar. So statement 1 is true. Statement 2, DNA evolved from RNA is more stable genetic material. Its double helical strands being complementary resist the changes by evolving repairing mechanisms. So both the statements are true here. Statement 1 and statement 2 both are correct. Option C will be our right answer over here. Next question you look into, ma. Given below are two statements, statement 1 and statement 2. Statement 1, tRNA or transfer RNA and ribosomal RNA do not interact with mRNA. Wrong. In translation process, during translation process, the mRNA after post-transcriptional changes to the mRNA at 5' end, first ribosomal RNA will come and attach, and after tRNA getting charged, transfer RNA will come and sit on the P subunit of ribosome, and it will copy the codons which are present on the mRNA. So for mRNA, both ribosomal RNA and transfer RNA, both of them will interact with the messenger RNA. So statement 1 is true. Statement 2, if you see, RNA interference or RNAi takes place in all eukaryotic organisms as a method of cellular defense. RNAi, which involves in the gene silencing technique or gene silencing method, is a well-known cellular defense method which is operated in all the eukaryotic organisms. So here if you see, statement 1 is wrong and statement 2 is correct, right? So here statement 1 is incorrect and 2 is correct. Option 2 goes right answer here. Next question if you see, ma, who proposed that the genetic code for amino acids should be made up of 3 nucleotides? Genetic code made up of amino acids with 3 nucleotides. It is given by the scientist George Gamow, that is option 3. Next question. Histones are enriched with what? Lysine and arginine. Okay, histones are enriched with lysine and arginine. Option C is right. So DNA is negatively charged, we all know that. During DNA packaging, the positively charged histone proteins will pack the DNA, and the major model we have learnt is nucleosome model. Okay, under DNA packaging we have learnt nucleosome model, wherein histone octamer, okay, H2A, H2B, H3, H4, 2 pairs of all these 4 histone proteins. Around this the DNA is wrapped out, okay, like beads on string. One DNA wrapping out this histone octamer and then moves to another histone octamer by linker DNA which is present, H2A, H2B, H3 and H4. So DNA is wrapped around this histone octamer, as such DNA is packed. DNA is negatively charged and histone proteins are positively charged. So around these two there is linker DNA with H1 histone proteins. Fine, right, ma'am. So this is about nucleosome model. And this histone proteins which are positively charged, they have the amino acids like lysine and arginine. This question you look into, which factor is important for termination of transcription? For termination process or termination step of transcription, rho factor is important. Sigma factor is for initiation. Sigma factor is for initiation of transcription. Rho factor is for termination. Here you see, this question has appeared in the year NEET 2024. Statement based again. This question is on Lac-Operon model. Okay, the question is on Lac-Operon model. Given below are two statements. Statement 1, if you see, in Lac-Operon, Z gene codes for beta galactosidase, which is primarily responsible for hydrolysis of lactose into galactose and glucose. Statement 2, if you see, in addition to lactose, glucose or galactose can also induce the Lac-Operon model. Okay, in addition to lactose, glucose and galactose also induces Lac-Operon model is wrong. So once you recall the Lac-Operon model, first is promoter, then operator, again a promoter region followed by 3 structural genes Z, Y, A. So Z gene codes for beta galactosidase. This enzyme will cleave the disaccharide lactose into glucose and galactose. If glucose and galactose are given to the bacterial cells, it will operate catabolic pathway, okay, which is called as catabolic repression. This will not be an inducer. Glucose or galactose are not inducers for Lac-Operon model, rather they are the repressors. Okay, so it will not induce. In addition to lactose, glucose and galactose can induce. No, glucose and galactose cannot induce Lac-Operon model. So here statement 1 is true, and 2 is false. Okay, next question. A transcriptional unit in DNA is defined primarily by 3 regions in DNA, and these are with respect to upstream and downstream. So here if you see upstream and downstream, what is upstream? Upstream is 5' end of the coding strand or the mRNA. Okay, 5' end of the mRNA or the coding strand is what? The upstream, and 3' end is downstream. This is upstream and downstream. In upstream, promoter is present, promoter followed by structural gene followed by a terminator. Promoter, structural gene and terminator will form up one unit, that is transcriptional unit. So what is the right answer over here, ma? Option 1. Promoter, structural gene and terminator will form out one transcriptional unit. This question you look into, match list 1 and list 2. Frederick Griffith has given us what? Transformation principle, who has worked out on Streptococcus pneumoniae. Francis Jacob and Monod has given Lac-Operon model, that is option 4. Hargobind Khorana has worked out on the genetic code after George Gamow, and Michelson and Stahl has given us the proof, experimental proof for semi-conservative method of DNA replication. So here if you have to look into the options correctly, ma, A is 3. Frederick Griffith worked on transformation experiment, A is 3, and B, Francis Jacob and Jack Monod on Lac-Operon model, B is 4. Hargobind Khorana on what? Hargobind Khorana on the genetic code, C is 1. Michelson and Stahl on 2. Okay, semi-conservative method of DNA replication. So C is the right answer. Next question. Which of the following statement is correct regarding the process of replication of E. coli? So, here if you see with respect to the replication of E. coli, semi-conservative method of replication only, no? Look into the all options, ma. The DNA dependent DNA polymerase catalyzes polymerization from 5 prime to 3 prime. Always do remember, polymerization is done by polymerase enzyme. In DNA replication, DNA polymerase is the enzyme which catalyzes the reaction. Always it will be from 5 prime to 3 prime direction only, and template is 3 prime 5 prime. At 3 prime the OH group is there, right? This is recognized by 5 prime, and the synthesis starts from 5 prime to 3 prime by DNA dependent DNA polymerase. So, statement 1 is true. Second, if you see, the DNA dependent DNA polymerase catalyzes polymerization in one direction, that is 3 to 5, wrong. Third statement, if you see, the DNA dependent RNA polymerase catalyzes polymerization in one direction, that is 5 prime to 3 prime direction. But the question is about replication, DNA replication. In DNA replication, nowhere there is an involvement of RNA polymerase, so we can eliminate this as well. Next, the DNA dependent DNA polymerase catalyzes polymerization in 5 prime to 3 prime as well as 3 to 5. No, polymerization is always from 5 prime to 3 prime direction. So, here the right answer will be option 1. This question you see, ma, which one is correct product of DNA dependent RNA polymerase? Okay, DNA dependent RNA polymerase. So, here it is given from 3 prime to 5 prime, means it is a template strand. We have to synthesize RNA strand because the enzyme is RNA polymerase. So, RNA strand is synthesized from 5 prime to 3 prime direction, from 5 prime to 3 prime direction. So, it is from 5 prime to 3 prime. This is an online session, beta, purely online. Yeah. So, this is from 5 prime to 3 prime. Here if you see at 3 prime and what is there? TAC. First you have to look into the complementarity. Thymine binds with what? Thymine binds with adenine. Adenine binds with uracil. Cytosine with guanine. Guanine with cytosine. Thymine is absent in RNA. In the place of thymine, you have to keep uracil. So, here if you see, it is A, U, G, here only we can write from 5 prime to 3 prime. A, U, G, C, C, G, so you can easily identify now. So, AUG, then UAC, AUG again. So, option 3 will be the right answer over here, beta. You also do parallelly, then you will get to know. Yeah, option 2 is the right answer. Okay. You also do parallelly, you will get to know. Option 2 is the right answer. So, based on the enzyme which is catalyzing, we can identify whether the question is about the synthesis of DNA strand or RNA strand. Based on that, it is easy to note down. So, when we looked into the all 2025 and 2024 NEET questions of molecular basis of inheritance, the questions are raised on what? Transcription processes mostly, and on replication, and on synthesis of the genetic strand, and based on the experiments and scientists. Okay, experiments and scientists and the model organism they have taken. And we have got the questions on transcriptional unit, lac operon model, on histone proteins. If we have to take a range, most of the questions have appeared on what, ma? Most of the questions have appeared on the transcription. okay, mostly on the transcription, replication and on the synthesis of new strands, and if it is match the following type of questions, we have got on the scientists and the model organisms and experiments. As such the questions are generated, right. So, in our coming class we will be looking into the next set of NEET previous year questionnaire on the same chapter, molecular basis of inheritance, by revising all the concepts as well. So, this will be highly useful to all the students who are appearing for NEET 2026 exam. So, that is all for today, Vita. Thank you.